So welcome. Thanks a lot for joining us this late, or I don't know what time is it in the other participants and our speaker time zones and so on. But yeah, welcome. And today we have actually quite a large representation of mycologists from North America. And as you maybe saw in our program, today we will have Jonathan Cazabonne, Mathias Rocheleau, and Harte Singer and Stefan Russell from different countries. I think, Jonathan, you are from University of Quebec, right? Mathias will represent mycology group Mycosphaera. Then we have Harte Singer from FunDive, which is the Fungal diversity survey in North America, and then we will have Stefan Russell who will talk about Mycomap project, right? And you are also one of the people that are how to say, they start the initiative of FunDive in North America. So welcome and thanks a lot for joining us and to be willing to talk about all of your efforts in fungal conservation in mycology in North America. So I think we can still give some minute to people joining, and then we can give the floor to Jonathan, right?
Yes, that's a good idea. I'll just very quickly explain the rules again for those who weren't here last time. Everybody's cameras are switched off and you're also muted. We do this to just keep the pace and not be interrupted by random noises. But you will all be given, of course, the opportunity to unmute yourself and show yourselves whenever we do the question rounds. At that time, know that this presentation is being recorded and we are going to upload it to the FunDive YouTube channel somewhere later this week. But if you do have any questions in the meantime, please put them in the chat and then we can circle back to them later.
Yes, and also last time our discussion were quite long, so this time we will try to focus only on answering the most relevant or important questions. And then obviously feel free to email us or our speakers, and you should find the contacts to all of us in Mycology Universe, I would say, or just in FunDive or FunDive websites and so on. Yeah, because I think today we will have quite tight schedule.
So hi everyone. Thanks so much for inviting me to give this short talk on fungal conservation in Canada, where I am happy to share where things currently stand, some of the challenges we are facing, and a few examples of efforts that are helping move the field forward. But before I begin, let me just briefly introduce myself. So as you mentioned, Marta, I'm Jonathan Cazabonne. I'm currently a PhD candidate in ecology and forest ecosystem management at the University of Quebec in Abitibi-Témiscamingue, where I mainly study the diversity ecology of soil fungi in old-growth boreal forest, mainly using eDNA tools. And I'm also the co-founder and the president of Mycosphaera, which is a Quebec-based non-profit organization working to advance fungal conservation in the province. But this is an initiative that Mathias will tell you more about right after my talk. And I'm also working as part of Team Laboule. I don't know if Danny has been able to join, but that's also part of my work. So with that, without further waiting, let's get started. So I will start directly with the actual state of fungal conservation in Canada. So here at the federal level, species can be legally protected under the Species at Risk Act, also known as the SARA. And as you can see in the figure, many groups of animals and plants are listed. There are also 29 lichens currently present, which is actually good news. But to date, no macrofungal species have been officially listed under SARA. And even though the legal definition of a wild species in the Act does not explicitly mention fungi, it defines wildlife species as any species of animals, plants, or other organisms, excluding bacteria and viruses. So by definition, macrofungi do fall under the scope of the Act. But in other words, the legal framework is there, but so far it hasn't been applied to macrofungi. So one of the very first steps to have some macrofungi legally protected is knowing which species occur in Canada, where they are geographically distributed, what threat they face, and what their risk of extinction might be. And under the SARA, it is specified that a general report on the status of wildlife species in Canada must be produced every 5 years, and this requirement is fulfilled through what are called the wild species reports. These reports began in 2000, and macrofungi were included for the first time in 2015. And in the most recent report, published in 2020, there was a major increase in scope with just under 7,000 macrofungal species that were evaluated and given conservation status using the ranking system developed by NatureServe. So this ranking system is similar in some ways to the one used by the International Union for Conservation of Nature, but it is specifically tailored to North America, and it assigns conservation ranks at three levels: global, national, and provincial, based on factors such as rarity, population trends, and known threats. And if we look at the results from the Wild Species 2020 report, we see that about 8% of all macrofungal species assessed are either critically imperiled or vulnerable. But at the same time, we see that around 75% of those macrofungal species assessed were either unrankable or not ranked at all. So in most cases, that means there was not enough data available to assign a conservation status with confidence. So we do know that a number of macrofungal species are at risk in Canada, but we also know that for the majority of macrofungi, we still lack the basic data needed to properly assess their conservation status. So to conclude this part, there is a huge lack of data on fungal diversity in Canada. We don't yet have a clear picture of how species are distributed within and among provinces, which ones are truly rare, what habitats they depend on, or how vulnerable they might be to decline. And in many cases, as we have seen, we also know far too little about their actual risk of extinction and the specific threat they face. And if I had to highlight just two keywords that really connects to the efforts I'm about to present in the next slides, there will be DNA-based specimen identification and citizen science. So now let's review some initiatives that are helping move fungal conservation forward in Canada. In the next slide, I will mainly show you, because of time, just two of the most recent ongoing efforts aimed at filling all these gaps in knowledge and conservation. But before that, I do want to briefly highlight a few milestones from the past last years. So first, the creation in 2021 of a working group on fungal conservation within the Canadian Institute of Ecology and Evolution. Some members of that group contributed to this 2022 publication that reviewed the state of fungal conservation in Canada, and where they proposed a list of 17 macrofungal species that could be prioritized for listing under SARA. And another key milestone was the establishment of the Canadian Fungal Research Network, also called CanFunNet, in 2019. And its primary goal is to connect researchers working on fungi across Canada, but also to bring together people also specifically working on macrofungi and help set conservation goals at the national scale. So for the first recent initiative, I want to talk about one I know best, the case of Quebec. So in June 2025, the non-profit organization Mycosphaera was created with the main goal of helping to document macrofungal diversity and advance fungal conservation in Quebec. This organization now has around 45 members and is mainly led by PhD students, citizen scientists, amateur mycologists, and researchers. And one of the main activities we do is conducting field inventories of macrofungi in ecosystems with high conservation value in Quebec. To give you just one example, I'm showing here one of the inventories we did last year in partnership with Nature Quebec, one of the largest conservation organizations in the province. This was in the Seigneurie de l'Aubinière forest, which contains one of the largest tracts of old-growth forest in the St. Lawrence lowlands, and it's planned to become a future protected area. And during these inventories, we have documented many rare species, some likely new to science, and others with conservation concern, just as this one, Rickenella swartzi, which is considered imperiled in Quebec. And I like using this inventory as an example because it also highlights the growing interest in fungi from some conservation organizations in Quebec and how fungal inventory data can be included in conservation decision making. Another recent milestone for the fungal conservation in Canada was a conference session held at the IUCN World Conservation Congress in Abu Dhabi in partnership with Nature Quebec and Parks Canada Agency. And this session had a focus on how to advance fungal conservation here in Quebec and Canada, and on what conservation organizations can do to support and help make that happen. And the last milestone for the province of Quebec comes from the end of 2024, when an open letter was published and signed by more than 70 researchers, mycologists, conservation professionals from Quebec, Canada, and beyond. And the goal was to urge the Quebec government to make fungal conservation a priority and to ensure that fungi are explicitly included in the provincial law that protects species at risk. Unfortunately, as you might expect, that's still not the case, but discussions have been open with the Quebec government, so hopefully one day we'll get there. And finally, the last example I'd like to mention is the Mycomap project that Stephen Russell will cover in detail in his presentation. This is a North America-wide initiative that documents macrofungal diversity using DNA barcoding specimens. And as of today, several Canadian provinces have joined this effort, including the Atlantic provinces, and very recently British Columbia has joined as well, and Alberta is coming on the board this year. And I want to specifically highlight the Mycomap BC project here, which is locally coordinated by Elora Adamson, because it's a really great model and an example of how coordinated efforts, community support, and also local capacity can truly help move things forward. And it's really encouraging for the future of fungal diversity mapping and conservation in Canada. And I'd like to finish by mentioning that there are active discussions happening across Canada about building more coordinated nationwide efforts to document and sequence fungal biodiversity. So I think we're going to see more and more projects emerging in the coming months and year to help move fungal conservation forward in a broader and more structured way in Canada. So I'm leaving you on that positive note. So thank you for your attention, and I'll be happy to take your questions at the end.
Thank you very much. I'm pretty sure there will be questions. So yeah, let's maybe continue now, right, with Mathias. So I will give the floor to Mathias, and then we can continue and finish with discussion. Thanks a lot.
Perfect. Thank you, Marta. I will share my screen. So I have more diapositive than Jonathan, so I will try to be quick. Maybe I will rush a little bit. So hi, I'm Mathias from Mycosphaera. I'm one of the co-founders and executive directors. So we co-founded Mycosphaera to promote the fungal biodiversity and help to recognize the fungal kingdom officially in the law. And the aim is that if we recognize the fungal kingdom in the law, it will bring financial leverage to studying them. And it can bring some innovation even for biotechnology, mycotourism, regenerative tourism. So we think that fungi could help the society a lot, and the conservation of fungi can be the first step to bring toward this. So first Canadian nonprofit fully dedicated to fungal conservation in Canada. Our 4 axes is explore, educate, preserve, and networking. So the main goal of Mycosphaera is scientific, educational, and for the long-term networking. So we want to do scientific inventories that we're already doing. Genetic sequencing, we will talk about this. We are not doing ourselves sequencing, but we are relying on Harte Singer and Stephen Russell actually. But we want to develop our own platform. conservation of specimen, creation of a fungarium. We do a lot of educational stuff like conference to be, we are in a primary school, college, secondary school, university, to make awareness of fungi happens. And the networking, we are building a full stack application that will bring all the mycology community together and hopefully try to reach with our Canadian myco enthusiasm. So the core team, because we have a big goal, we need some competent people. So you see that it's the core team of the Mycosphaera. So we have a scientific board, mainly ruled by a PhD candidate in chemistry and forest ecology. We have a mycology teacher. We have a museum candidate, a PhD museum candidate. We have mycological representatives, bioinformaticians, people from finance. It's very important for us, and people from institutional organization like Laval University, Biopter, that is a research center, and national research, the ENRS, that is a national research center, scientific center. So we like to bring art and science together. It's one way that we think it's innovative. So we have built a theatrical mycology conference. So that is provided to family. So we want to reach people at a young as they can. To make people more curious about nature. I think mycology is a good vector to knowledge transmission and curiosity. And because we are lacking of mycologists actually in Quebec and Canada, because it's not something that you can learn at school. So by reaching them as young as they could be like 4 years old at those theatrical mycology conference. So I think it's a good way to try to reach more people. We have a museum exhibition that have been built by our PhD student in art conservative and museology. So we are preparing a second exhibition in Université Laval, and we are talking with a regional museum to make a itinerary museum exhibition. And we hope that we can bring this at a bigger scale, maybe that could move in other museums across the country or the province. We do a lot of macro photography. We have 2 people specialized in macro photography, myself and François Gay. We won the national scientific contest of photography. So it is a new species for science that has been discovered 2 years ago. So we use the photography aspect to leverage the awareness of fungal biodiversity. And something about Quebec that Mycosphaera is related, but Quebec, we had a delegation of Spain from the European Mycological Institute last September that provide the first label in North America from the fungi-friendly forest, because Kamouraska has been recognized for its mycotourism aspect. We have to know that Spain was one of the international leader of mycotourism. We have been invited to International Congress of Mycotourism to give a talk there last year. So Kamouraska won this label, and we have a research center, Biopter. It's a mycology research center, applied mycology research center, quite unique in Canada actually. So we can see what the research center do. They are researching aquatic fungi from the St. Lawrence River. And we made a partnership with this research center. We applied for a fund with the National Research Fund of Quebec to discover new species of fungi in aquatic St. Lawrence River. And actually, they discovered some entomopathogenic fungi in the St. Lawrence River in sediments. We were in the International Mycotourism Congress in Soria Gastronomica. to talk about how the Quebec has the aspect to promote the citizen science and can leverage the mycotourism aspect. Like I said, Spain is one of the leaders of the mycotourism, but they were here to try to know more about Quebec initiative and the aspect of Quebec that Biopter is a research center in applied mycology. They were interesting to bring some citizen science and mycotechnology aspect in their mycotourism definition in Soria Gastronomica in Spain. So it's a good milestone for us to have been invited by the European Mycological Institute. We have a partnership with Biopter, like I said, and we applied for a Genome Quebec grant to develop the nanopore sequencing, as Stefan Russell and Art Singer are developing. So it's pending. We will have news like in 1 month. And like Jonathan said, fungal biodiversity survey, we are making some reports. So they are quite different from like citizen science approach. It is citizen science approach, but we need to be more sophisticated. I would say that we use geometric data and permanent station to assess the biodiversity survey because it's a federal provincial reports, and we need to do a little bit more statistic to do those kind of survey. And I will tell you more. I'm ending my presentation on that what's our pipeline actually, and we want to be in an approach that help the citizen science freely, and approach that it's more an internal approach for those kind of biodiversity survey. So standards and protocol, like this study that has been published in 2012, monitoring fungal biodiversity towards an integrated approach. I think Jacob, if it's here, one of the co-authors. So the goal and the difficulties is the database are spread everywhere. It's not well structured, and often the data are lacking of standardized data. So our goal is to make it more easier for our organization and for the citizen science people. So we have a geometric approach. We standardize fields, so we have a relation table in database that are not textual, but they are standardized. So we use 3-liter code automation trigger that people will use INAT on the field, like a survey, but it will automate a trigger. So we use code to avoid like misspell words. It is an example of our database that it's made on SQL Notion database, but we have standardized the plants. We have like 4,000 plants in our database. In Quebec, we have standardized the 45 eco-forestry type. We have standardized the substrate, the habitat, even the morphological aspect of the mushroom. We have standardized the things. Our goal is to make an integrated dashboard, is internal dashboard for now, but people that could create project. And here we can see like all the all inventory outflow register, sampling station. So we do sampling station. So when you do a geographical set of mushroom observation, if you are in the station, it will trigger an optimization that will bring the plants because we have already did an inventory of plants. Uniformity of habitat and substrate. So we have created a database for those. So we can watch all the observations that have been made with those kind of substrate or habitat. And we have a reference list of taxa that are associated to those database. We have a taxonomic group feature too that we have standardized. And this is a pipeline. We use INAT at the start. We aggregate the compilation of standardized data on our database application, and we push it on INAT. Is very important for us. So our process is to make more quality data during the middle. And our application is a Fungarium manager application. So because we were thinking about why people will do their own Excel CSV on their own with unstructured column. why not to bring one fungarium database that the structured data will be given to them. So our next milestone is to develop the nanopore sequencing in Canada, in Quebec, and to unify a portal that will bring genomic observation, program manager, referential taxa, teaching resource, scientist portal, and mycotourism portal. And we are working on a predictive map that will, here is the forest data table from that brings ecological data from the forest, the soil, and the perturbation of the forest. And each observation could bring those data to the observation. So even if people don't collect data from the ecological part, we can stick metadata to each observation. So it could help a lot to make some predictive map of if we see elm trees, which mushrooms are linked to elm trees, and we could search for predictive search. Thank you. And maybe I could share something that we are building is our application right now. It's the association of 2,000 mushrooms and 100 plants. And the red link are saprophytes, parasite, the yellow, the saprophytes, and the green is the symbiotic. So we can see the trees are related to mushrooms, and you can see each mushrooms are related to each trees, and you can search by name of by taxonomic group to see all the relationship from mushrooms. So it's a funny way to see the galactical way to see the relationship between mushrooms and plants. So it's, I think we are building something that could standardize the association between the mushrooms and plants.
Thank you very much. That's very, very, very cool. And I think we can go to Harte's talk, and then all of the questions will again go for the end, right? Thanks, Mathias. And I think I can give the screen to Harte.
Great, thank you. Can everybody see my screen?
Yes.
Great. Okay, so I'll briefly introduce myself. My name is Harte Singer. I currently am a student at Cal State East Bay in Hayward. I'm a master's student studying fungal endophytes. I got my start getting into mushrooms as a chef. That was most of my life was spent in the restaurant career. And then I got so into the mushrooms that I left the kitchen and began focusing on mushrooms. I currently am the sequencing program director for the Fungal Diversity Survey, or FunDis. I also am on the board of a few nonprofits, local mycology organizations. I also run a private company called Dicaria LLC, where I do specimen barcoding of fungi. Fungal Diversity Survey has been sort of the core organization that I've been associated with recently. So I'll talk a little bit about that. We go by FunDis for short, a lot quicker to say. And we have a vision in which the fungal kingdom is fully documented, understood, appreciated, and protected. And then FunDis is a group of community scientists who share that vision. I'm going to use the term community science. It's the same as citizen science. It's just a little bit, more inclusive, I guess. So what does FunDis do? So FunDis runs a couple of programs, maybe the most famous of which is the Rare Fungi Challenges. These are programs that engage the public to seek out rare and poorly documented species. We put together lists of these fungi. Here's an example of one, Mycota luteophylla. To this day, this specific mushroom has only been found in one location in California, under one tree. So we suspect it's a single individual, has never been found outside of that. There's some similar looking species. So we challenge people to go look for these and post their observations on iNaturalist. And these studies run for a period of time, at which point, at the end of that period of time, we can assess, okay, which of these things are truly rare, which have just been underdocumented, and then we reevaluate and add different species. There are 4 challenges right now. There's the Northeast, the Southeast, the Rocky Mountain, and the West Coast Rare Challenge. The other main project that we've been doing is the California Fungal Diversity Survey Project. I'll talk about that in the next couple of slides. And then we recently hired a conservation director who's going to directly connect the work that we're doing with organizations like NatureServe and IUCN, so we can begin listing these species that are truly rare. So the California Fundus Project, this is really awesome opportunity. It's based in the what's called the 30 by 30 initiative. This is something that was passed by the state legislature to protect 30% of all the land in California by the year 2030. And one of the components of this project is to establish an inventory of all the taxa in California. So that's the all taxa biological inventory. And the funding from that, some of it went to an organization called the California Institute for Biodiversity, and that is the organization that gave money to Fundus to do this effort. Entirely community science led. So the people that were hired for this were not like PhD mycologists, or even people, some people didn't have bachelor's degrees. It was really just folks who had been active in the community in California. A lot of people's resume was their iNaturalist account, and you can see really quickly how much people know about fungi just by looking at their iNaturalist. And we decided to establish a complete field to fungarium pipeline for this. So really a completely closed system where every element of the process is taking place under sort of one umbrella, and that allows the results to be completed rapidly. So I'll just go over this as like a workflow. So the beginning of this is paid collectors who have a ton of experience going out to the field, going to a variety of habitats. California is extremely rich in different plant habitats, and so people with specialized knowledge in our in our habitats went out there, made collections in the fields. The photographs they upload to iNaturalist. I'll go over that in a second. And then the collectors would dry the samples and collect a tiny piece of tissue, put it in a PCR tube, and send it over to Cal State East Bay, where we had taken over a lab bench to do the DNA barcoding. All the specimens were then accessioned in the fungarium at Cal State East Bay, with a select few going to UCLA and Los Angeles, specifically the collections that are from around the Los Angeles area. And then the sequences were looked at by a team of paid employees who would look at the raw data, look at the taxonomic assignment, and really make decisions on what to do with this. The advantage to this is that we can control every aspect of it. We can control for costs. Originally, we were using University of Guelph to do sequencing. It was very expensive. So it really allowed us unprecedented control over the entire process, which was great. So I mentioned iNaturalist, and so did Mathias. So this is the primary community science platform in the United States that's used in the mycology community. There are a few others like Mushroom Observer, but this is really where all of the action is going on nowadays. It's integral to all of our workflows, and it allows anybody to make observations and contribute to community science. It's available with a smartphone app as well as a website, and it's very approachable. I'll go over the anatomy of an iNaturalist observation, and I have a few slides on this, but maybe I'll just do it live. because that might be a little bit easier. So can you all see this screen?
Yes.
Yes.
Great, excellent. So here's an example of one of our observations. It's held inside of a project. This is the total number of observations made for the California Fundus project. And I can use some tools like in the URL to filter just the ones that have DNA barcodes. So a lot of the newer collections, we are not barcoding as we don't have funding to do it. So we only have 10,000 barcodes for the 13,000 collections. Here's a single collection. It shows coordinates, and the coordinates are, you can see in some cases it's the accuracy is recorded, but the user could obscure those coordinates if it's particularly sensitive taxon or if they want to protect their privacy. But I can access all of the true coordinates as the project manager. The observer would take a high quality photograph showing all the features of the mushroom, another photograph using a standard field tag with a unique number, a QR code for easy scanning, and a scale bar. And then we would instruct the collectors to take pretty detailed notes. We want the abundance of the fruit body. We want the associated species. So just like Mathias was talking about, how important the relationship is between plants and fungi, we had them record basically the top 3 or 4 plant species that were nearby. So this is Douglas fir and Kenyan live oak, and there's a few more at the bottom. This was DNA barcoded, so we actually put the DNA barcode sequence right here. We have the location. We have what we call a provisional species name. I'll get into that in a minute. The tag number, etc. And then also linked here is a link to the BLAST results. And so this is using the Mycomap platform that Steve will probably talk about a bit. And this allows anybody to see the thing that the person who analyzed the sequence was looking at. And I'm not going to go into detail about how to interpret these results, but it uses both the NCBI database as well as a local database that includes many other sequences that are not on NCBI. You can access the sequence data here. I can download just with this link the FASTQ file right to my computer. If I want to double check the 497 reads that were used to create this, I can do that. And then another link takes you here to the location in the fungarium. So I can very easily and quickly request this, this collection from Brian Perry. I'm going to move forward and talk about some of the strategies and workflows that are used in community science in the United States. A lot of us are going under the flag of this organization called the North American Mycological Association, and we have our committee that's just DNAMA. And Stephen Russell is part of this. A number of other folks who are doing the on-the-ground work in the United States are part of this group. I had mentioned before that there's a process called sequence validation. So this is one of the primary goals of this organization, is to standardize the approach that we use to determine if the sequence is real, does it match the specimen that's pictured, and does it represent the raw data accurately? And so through this, we train volunteers to use the publicly available platforms and tools to analyze DNA sequence data and try to establish consistent process and best practices. The main goal of all of this establishment of consistent processes and best practices is to eventually get to the point, which is coming soon, where we can partially automate the process of assigning a name to a sequence specimen. And so you can see here, this is taken from Stephen Russell's mycomap.org dashboard, which is currently under development. And it shows the total number of unique taxa that we have DNA barcoded. It shows how many temporary code names we've developed. And then importantly, it also shows the species rarity distribution as far as derived from sequences, there's only 385 that are very common. 2,000 that are common. You start to get over here, and with collections that only have 2 to 4 observations, that makes up the entirety of the other ones. And then what makes up more than everything else combined is what we call very rare, which only has a single record as determined by a DNA barcode. So, we want to be careful in saying that these are all new species or they're all rare species. The line between species is one that is you have to carefully determine. So based on the results of DNA sequencing, this is how many we have found that only occur once. And that harkens back to the difficulty it is that there is in actually listing these, because if you only have a single collection, you can't list it as a rare species. Okay, how we do the tracking of this work currently is using a Google Sheet. It's maybe not the most elegant method, so we're in the process of developing a more bespoke dashboard, but you can see the people who are working on it, put their name here, decide if the sequence is good, is it contaminated, did it fail, and then they can put whether they validated it. You can flag something for review, and then we assign our names and we put notes and track various metrics. So I want to get into the temporary code name. You saw that on the Quaternarius observation. This is a system that we have been using. It's not like we developed this. This has been used by various different people in isolation, but what we're trying to do is all agree on the use of these names. So the correct term for this is a temporary code name. Some places you'll see them called provisional names. That's not really the correct usage of the term provisional name, so we're transitioning really to using temp code or temporary code name. And this allows us to communicate about taxa that have no reference barcode data and can't be reliably identified to species morphologically. The new taxa are being discovered much faster than we can describe new species, so this is really a necessary thing. In the past, people had just said, oh, Agaricus spa or Agaricus af, bisporus. Now we can use a series of letters and numbers to identify these things, so that every time somebody sequences one that matches this, you at least have some name to give it. The beauty of this is that it's not held down by the nomenclatural code. It's more anarchic, and it allows, the whole purpose of this is to be temporary, so there's no need to necessarily adopt a system that is precisely shared by everybody. So you can include this format or this format, as long as we all agree to use the same name for the same molecularly derived taxon. Some other sort of workflows. There's the commercial MycoBlitz, which happened recently. And this was a really cool initiative that had volunteers send in mushrooms that they found at the grocery store, so that we could try to see, do the names match what they're supposed to be, or really what are they? So many of them had no labels. And for example, many of the porcini that we found were just Swillus luteus. So kind of a really cool way to engage the community and also begin to establish truth behind some of these sort of random mushroom products. The main initiative I want to talk about in workflow is the 4A sequencing. So this is something that Stephen Russell began doing in 2017 or 2014, if I remember correctly. And it involved sequencing all the specimens collected at a large 4A event. We started doing this more as something that was funded and organized in 2024 in Randall, Washington. And it's bringing the entire pipeline under one roof. So the general public goes out and collects mushrooms for 4As. These are generally people who are inexperienced. Most people are not using iNaturalist. So we're really just using the 4A as a way of gathering all the specimens. And then we do the PCR and sequencing on site. From these events, we've basically got a 2 to 6% new taxon discovery rate. And I say new in quotations because we can't really prove something is new until we have all of the holotype sequences, which is an ongoing process. Here's how the workflow goes. You have the field slip, which has a unique identifier. We have some stickers that indicate the sample number. So this is which PCR tube it's sampled into. And then a small portion of this specimen will be saved as a DNA voucher, which will usually include hymenium. So someone could do microscopy or do repeat sequencing. It's enough of the starting material to have as a reference, but not as something that you would describe the species from. The way that we do the sampling is I use individually wrapped toothpicks, dip it into the hymenium. You get a very, very tiny piece of material on there, and then it goes into a buffer. This buffer is really easy to make. It's extremely effective at preserving DNA. I've had samples sitting in this buffer for 6 months at room temperature that still generated great DNA sequences. Then that goes to DNA extraction, which I just had volunteers do. I trained people who had never done PCR before to do this. Although this guy has done a lot of PCR, we're not going to look at him. And then sequencing on site. So oftentimes the nanopore sequencer takes a long time. So I had to leave the event with the sequencer still running. And then this goes to a team of volunteers for the sequence validation. And that's all I have for you guys today. So I'll pass it along to Stephen Russell.
Thanks a lot. And yes, we can continue with Stephen. So thanks again.
Hello, everyone. Can you see my screen?
Yes, we can.
Yes.
Okay, great. So hello, and thanks for inviting me. My name is Steve Russell. I'm the director of Mycota Lab. And so our lab is a nonprofit research lab based just outside of Ann Arbor, Michigan. I'm coming to you today currently from the University of Michigan, so that's my day job. All of my work within mycology is actually through our lab, which is not affiliated with the university. And currently I do it all from a volunteer perspective. And so some other unique aspects of Mycota Lab. We started in 2015, actually. For most of that time, up until about 2024, it was a small home-based lab. That's where we got our start, and that's how it progressed for almost a decade. It was entirely an at-home endeavor. Another unique component about our lab is we conduct collections-based research. We get lots of collections, as we'll talk about in a minute, but we're not hypothesis-driven. So primarily, we're interested in building foundational data sets. We don't do much publication in the peer-reviewed literature. We're really focused on data generation at this moment in time. We might have more of a focus on that in the future, but probably not in the near term. But I would say that's probably a pretty unique component of our lab, especially relative to academic labs, is not being hypothesis-driven. Another unique element, virtually all of our collections come from community science around the country that we'll talk about in a moment, but our focus is really on high throughput specimen collection and sequencing. We have a really intense focus on process and methods and automation, all in the service of cost reduction. And so one of our primary metrics that we think about often is how much is the cost per specimen for our lab to actually generate. And that's really one of the key things that we try to drive down over the course of time. So let me talk to you briefly about some of our primary initiatives. Our primary one would probably be the Mycomap network. And so back in 2015, we started a project in Indiana to start to document all of the biodiversity that exists in that state. I don't know if you guys can see my cursor, but Indiana is this green area here right in the middle, right below the Great Lakes in the United States. And we started off really small at that time, but our goal was to begin documenting all of the macrofungi that exist in Indiana. And what quickly became obvious to us was that if we were going to do that successfully, we really needed to start a system to get DNA sequences for all of the specimens that we were encountering. And so really starting in 2017 is when we started the DNA work. at the statewide level. And it kept on growing year after year. And eventually in 2023, we started opening up to beyond the one single state of Indiana into more of a continental scale effort. And so that was starting in 2023. At this point in time, there are probably around 20 states and provinces in North America, to where anybody that lives in that state can send us any collection that they find at any point in the year. And so if somebody's in the state of Colorado and, you know, it's July and they go collect 100 specimens, it's fine for them to send us all 100 of those collections, and we'll do DNA barcoding of those specimens. And so that's true for all of the areas that are highlighted on the map in the lower left. We'll give one quick project shout out. I mentioned I now live in Michigan. I spent most of my life in Indiana. I've lived in Michigan for about 3 years. But since 2023 is when we started the Michigan project, we've been able to DNA barcode 11,000 observations. And this picture is kind of small, but we've been able to get a pretty good coverage all across the state. And it's a pretty large state. And so these kind of efforts are our primary focus. At this point in time, we probably, our lab, it's a small lab. We only have one full-time employee. Processes this year, I'm guessing well over 30,000, probably approaching 40,000 or more specimens per year. But for the last couple of years in 2023, in 2023 and 2024, we're averaging, you know, 25 to 30,000 specimens a year. And so it's a really substantive effort overall. We get lots of specimens coming in. And we store all of these specimens in our herbarium. We have a herbarium called Myco that now has roughly 100,000 specimens in it. Another one of our major initiatives is called the Continental Myco Blitz. So 3 times a year, the winter, the summer, and the fall, we have people from around the country. If we go back to the previous slide for a second, all of these collection efforts during the year are limited to states that are highlighted here. These are our target areas at the moment. But during the Continental Myco Blitz week or weeks, anyone from anywhere in the continent can submit specimens to our lab. And so that's several weeks a year. Thus far, if you see on the left, there have been about 61,000 observations made. I think roughly about half of those have specimens associated with them. So these events alone, just over the last few years, have probably brought us in the neighborhood of about 30,000 specimens into our fungarium. So we plan on holding these in perpetuity, long into the future, 3 times a year, winter, summer, and fall Continental Myco Blitz. About 1,300 people have contributed so far to this. For both of these projects, one of our core goals is to try to meet people where they are in terms of their ability to access the projects and to participate. And so one of the ways we do that is by making the protocols as simple as possible. And so in order to send stuff to our lab, all people have to do is to document the mushroom on iNaturalist, dry it out in a dehydrator, and mail it in for DNA. Just this year, we're adding one new component, which is printing off a label that has a QR code, because that really helps us to expedite check-in. But we really, that's at its core, that's as simple as our protocols are. Is to document it on iNaturalist, dry it out, mail it in. And we want to try to keep them that simple just to encourage participation. There's a lot more protocols that people can read on our website. You can check out mycomap.org. We just recently published a development site that I'll talk about in a second that has the full set of protocols, but they're really just additional details or additional considerations for power users, for people that want to go out and collect 100 specimens and organize them in an efficient way and get them to us in an efficient way. But for the people that are just going out and collecting 3 or 5 things, we try to keep the protocols as simple as possible. We have a lot of other work and interests. We recently completed a fairly large fungarium sequencing project where we developed a lot of new workflows and methods for it, primarily based on nanopore sequencing. We're currently working on a type specimen project as well. Both of these are focused on the University of Michigan, although the types, we're actually sequencing all the types at the Field Museum as well. If you're familiar with the University of Michigan, it's actually one of the reasons why I ended up in Michigan. Without this fungarium being here, I doubt I would have moved to Michigan, to tell you the truth. But we recently completed a survey of 6,000 specimens from California, from the state of California, that are located at the University of Michigan. And so I believe it's probably, at least as far as I'm aware of, the largest fungarium barcoding project to date. And we were able to get about a 70% success rate with those specimens with the workflows that we developed. The median age on those specimens was about 75 years old. And so, you know, if you look at the next largest fungarium project, it was done with Sanger, and they had about a 20% success rate on those specimens. And so using next-gen, it doesn't have to be nanopore, it could be one of the other next-gen technologies as well. But next-gen is a really vital component for anyone considering kind of large-scale DNA barcoding of museum specimens for a bunch of different reasons that I don't have time to go into today. We do a bit of culturing in our lab. We just finished up a culturing project. We don't focus on that too much, but that was a fun one for one of our lab members. And we're also really interested in not just the lab side, but also the bioinformatics side. And so to be able to process this data effectively and to be able to continue scaling the data in the ways that we need to, it became clear to us pretty early on that most of the bioinformatic tools that were available were less than stellar. And so we actually went to the drawing board and developed, at this point, we have in our current implementation of our website 4 different novel bioinformatic tools for everything for the entire process flow, from sample demultiplexing to species clustering to blast interpretation. And so we've been really trying to go back and think of what we need to do from first principles. And I'll tell you, when you get under the hood of a lot of the bioinformatic tools that are out there, you see a lot of deficiencies really, really quickly. And so that's another reason why, you know, we've really put a focus on bioinformatics. And I would say all of these tools have been the result of another citizen science collaboration, community science collaboration with other individuals. We're just communicating through a Facebook group chat about our needs and about you know, what needs to be prioritized, what's working, what's not working with these different developments. And one of the really cool things about it now with AI, it's all AI driven. So we can have somebody developing a tool such as the one you see on the right called GapHack to be able to generate a barcode gap analysis for a set of blast results. And so somebody could generate it one day, and then that same day I can have it implemented into a live install with live data, fully integrated into our framework. And so AI has really been able to take the turnaround time for some of these bioinformatic tools significantly lower. This is the period of time in software development that I've been waiting for my whole life personally. And so it's a really, really fun time to be able to use these tools and to play with them and explore their capabilities. Just a shift in the background coloration. The final few slides, I'll talk about the Mycomap webpage. Just one more note on bioinformatics. This is an example of what we call mycoblast. So our local blast results. And this incorporates a lot of the tools that you just saw on the last page, like the adjusted identity calculation. So instead of there being an alignment, so if you see on that 4th line down, there's a drop down to where you can see the alignment, and it will automatically adjust the percent similarity associated with a record compared to what NCBI calculates, you know, based on are there homopolymer repeats, are there ambiguous nucleotides, and it will autocorrect the identity value. And when you do that, the barcode gap analysis often becomes much cleaner. And the barcode gap is just more typically easier to spot visually once these corrections for common sequencing errors or common blast deficiencies are incorporated into the analysis. We've been doing a lot of work in terms of collecting data that I talked about, but one of the key things in the future for our lab is going to be interpreting the data. And we've purposefully put off any type of sustained conservation efforts for our lab, just so we can focus on collecting the data. But at this point in time, we're starting to have enough data to be able to look at these large scales and really narrow scales, individual states, have enough data to start interpreting that data in meaningful ways. And since we're so focused on community science, it seemed like one of the first steps into fungal conservation should probably be a community science initiative. And that's what you're seeing here with the certified fungal habitat. Just in brief, the first step is a residential program. We're likely to have a program for land managers in the future. But in brief, the landowner would download a checklist of conservation actions that they could take. They would get points for different types of actions, depending on the complexity of the action and the importance of the action. Once they reach a certain amount of points, they would be able to put in an application to have a certified fungal habitat for their residents. And so, I kind of refer to this as the need here is as much marketing as anything else, because the main way that a program like this is going to succeed and to actually have an impact is by lots and lots of people doing it, right? The way to get real conservation outcomes from a program like this is to have lots of fungal habitats all across the country. And so this is probably something, especially for the people here in the United States, you'll probably be hearing me talk a lot about this for the foreseeable future. But this is one of our primary initiatives this year. And I'll go ahead and wrap it up just with a couple thoughts on the future. I mentioned mycomap.org as the website that we're prepping to launch at this moment. If you've explored Mycomap or seen the blast links in the past, it probably took you to the .org site that you see in the upper right, or sorry, the .com, mycomap.com site that you see in the upper right, the blue and white site with the title of Mycomap Beta. I actually looked it up the other day. I actually bought that domain name in 2014. And so in my mind, I've been working on it since about 2015. But in that whole time, I've never taken the beta name off of that site. We've never installed a nice looking template. That site was never really meant to be public facing. It was meant for us as a test bed to be able to develop a lot of cool functionality, to understand our workflow needs. And it's used currently by 150 DNA sequence validators, week in and week out to organize projects. But it was never really meant to be public facing. We never developed usage documents. Everyone who's been trained on it has really undergone personalized training. What we're launching now is a new website called mycomap.org, and it's meant to be very much the opposite of that. It's meant to be public facing. It's meant to be really easy to find data. It's meant to be really easy to look up species names, to find all of the observations associated with the temp codes Harte talked about. And we're calling it a research node. If you look at the panel to the lower left, it will give lots of summary statistics, things like which states have the most global first records that have been discovered. They'll have an activity feed of essentially real time as new species are discovered. They would pop up in your activity feed. We have species accumulation curves that are live, based on the current data. And if we do lots of our communication through the Facebook groups, Fungal Sequencing and Fungal Sequencing Results. You can see a full version of this. I posted it earlier today there. But that's where we do a lot of the communication. But this is the launch of this website, as well as the Certified Fungal Habitat Program is really one of our labs, a couple of our lab's core goals this year. But probably mid-year, we're going to start on another platform, which would be the secondary goal, is to really start to try to integrate the above ground and below ground data in a meaningful way. So I have a keen interest in environmental DNA and soil sampling, and really all, not just soil, but all types of environmental DNA. And really integrating the above ground and below ground in a meaningful way is really going to be one of our top priorities for this year, as well as fundamentally changing the cost curve of collecting and sequencing eDNA samples. Environmental DNA research is never really going to be able to scale the way it needs to be able to scale for us to be able to get the answers we want if the cost structures are the way they are. It's just not sustainable. It's not scalable. With Nanopore, we were able to go from $10, $15 per specimen that we were paying for Sanger. Right now with Nanopore, we're paying roughly 20 cents per specimen. We need that same kind of multiple order of magnitude decrease in the cost structure for environmental DNA samples. And that's something I think we have a pretty good beat on already. And I think that we'll be able to do this year. But for us, we want to be able to have scaled, large scale, continental scale environmental DNA efforts. And the only way that's going to be feasible is by fundamentally changing the cost structure of environmental DNA work. And so with that, looks like it's time for me. So I can open it up for questions.
Thank you very much, and thank you all for presenting this great talks. And I'm pretty sure there will be plenty of questions. So if someone would like to ask something, please post on the chat, and you can also specify to who is the question. But if you don't mind, I don't know if there's anyone else who would like to start the discussion, but I might have some questions. So maybe I can let myself to start. So it's like it's all very inspiring, and I was thinking during your presentations what we can do, for example, in Poland, how we can enhance our mycological, our efforts for mycology in general. But I was thinking more about how you actually maintain all of this work and these jobs with, for example, self-funding, or the funding that is not really coming regularly and continuously. So I have one question to Harte. The one is specifically dedicated to the sequencing method, like to DNA extraction method that you are doing within the events that you are organizing. So my question would be, what is actually you are using? So when you are taking, when you are touching the fresh specimen with this stick, and then you go into the buffer, what is this? Is this what method are you using? Is this super efficient, or are there more challenging species of fungi that are not as easy as the others? And what is this? Is this Chalex, as we are using, or what is the maybe not really detailed protocol, but
great question. Yeah, so it's extremely effective. The raw success rates from some of these forays get up into 95% range. If it's done really well and people are following instructions, that is, of course, the challenge. Like anything else, instructions can be misinterpreted. And so the key features here is that you are taking very little tissue. You saw how much was on the toothpick. A lot of our failures came from people taking too much tissue and overwhelming the buffer. The buffer itself is very, very simple. It's an old recipe that was, we're not sure where it came from, but it's been used by a lot of labs, and we're calling it XAMP is our sort of name for it. It's EDTA, tris buffer, potassium chloride, and the pH is adjusted to be just above 9. And that is it. It is the simplest thing to make. We have protocols on Stephen's protocols.io and my protocols.io. Maybe Stephen, if you could shoot a link to that.
Yeah, I just popped it in the
it's
chat.
already shared. Yeah. Thank
Great.
you very much. Yeah, I think we used that at least in the beginning of FunDive, but I think the problem were that we might use too much of the fresh material because the extractions were not as efficient. So maybe that was the problem. Yeah, thanks. That's really useful. Yeah.
Yeah, it can be terrifying when you're doing it because you don't see anything and you say, oh, it's not going to work. But those are the ones that work the best. So yeah, give it a shot. And if you ever need, more help with that, I'm happy to provide it.
I would
Well,
just add there.
thanks a lot.
Yeah, I would just add that we use that protocol and that extraction solution for all of our sequencing work. And it also works really well for a lot of non-complex eDNA samples. And so the one addition that we might add to it in various circumstances is just putting a small steel bead in there and bead beading as well. But yeah, as Harte mentioned, using the smallest amount of tissue conceivably possible. If you can easily cut it in half with a pair of tweezers, that's too much tissue, right? You want to use the smallest conceivable amount of tissue that you can grab.
Okay, yeah, that makes sense. Also, I remember with Irina that I see she's present here was also using very, very, very little piece of, actually it was a slime mold, but it was very efficient. And okay, thanks a lot. And I see that there's at least one more question from Jakob because Jakob is raising his hand, right? Or can we unmute Jakob or should he? I don't know, Glenn, if that's possible or maybe, yeah, 1 second.
Yes, Jakob, you can go. Sorry.
I think I'm disappearing here. So
Yes.
amazing insights into mycology in North America with a strong community science perspective. I'm really fascinating by what you're actually doing and achieving. And I mean, I have so many questions, but to give you a very overall question, I'm very curious to how you see the kind of long-term sustainable funding schemes for what you're doing. I mean, you're doing different things, but in Europe we now have the FunDive funding. It's running out in 1.5 years. Some even within half a year. It's not easy to get the new funding. So we need to build up long-term systems, and it's complicated in a continent like Europe where we have many different nations. And I'm just very curious to hear what kind of solutions have you found for that?
I could answer briefly, and I'm sure there'll be other folks that want to answer. You bring up an excellent point, and the funding that we got for the California Fundus project this past year was about 10% of the funding that we got the year prior. So we've had to, completely contract the work that we're doing and go down to a skeleton crew, and it's not ideal. The part of the reason why I started a private company is so that I could fund my own work. And so, built into the cost of doing other people's work is enough for me to satisfy myself. But it's a really huge problem establishing funding. And I think Stephen and I have done a lot of talking about how we can do this in North America, but a lot of talk so far. We don't have a lot of money coming in, but maybe Stephen has some more context for that.
Yeah, I mean, I would just say we've been trying to get funding for, this type of work, in a sustainable way since I started a decade ago, right? And we've really never been able to find a consistent and long-term methodology to fund it. Most of our work is funded through local clubs that have an interest in doing the sequencing themselves. So, they would give us X amount of dollars every year for, X amount of specimens. But there's not a huge number of clubs, able to do something like that. And so that kind of, and even if we maxed all of them out, that would still be a relatively low number at the end of the day, especially from like an academic grant perspective. And so that's really why our focus as a lab has been on fundamentally changing these cost structures to where now we're able to decrease, to have a 2 order of magnitude decrease in the cost per specimen. And that's how we're able to scale our projects without getting more funding or without additional funding. It's really been the focus on that cost basis. And so we're able to do exponentially amount of funding that we were just a few years ago.
And I would add, as a nonprofit startup that just started last year, so we see the bottleneck of funding because partners will not give grants to small and a new organization that doesn't provide like financial states yet. But we achieved some grant from our environmental organization that are not related to fungal kingdom, but they see fungal biodiversity as an innovative way. So it's we tap on the speech of this is innovative, and actually we are reaching with Genome Quebec. This is a part of Genome Canada institution. So they see it like an innovative way. So I think the innovation patterns could leverage some grants that are related to innovation. And we are trying to tap on the innovation tech here, because in Quebec we don't have any organization doesn't provide nanopore sequencing inside Quebec. So we try to tap in the innovative tech grant to achieve those kind of grant, and we try to achieve some philanthropy international funds too. So it's tough. It's not a free game, but I think mushrooms has something to get close to those grants. Yeah.
If I could just mention one more component in brief, one of the things we haven't tried that we're trying this year is a large-scale crowdfunding effort. And so the new Mycomap website has a lot of functionality that becomes unlocked when people join a membership. They would still have access to all the data at any point in time, but they get additional features, additional types of analysis if they sign up for a membership. And hopefully there's enough people that are just interested in the work overall to help support it. But trying to do that on a large scale is something we haven't tried, but we are starting this year.
Super interesting. Yeah.
Thanks a lot for the discussion. So I don't know if there are more questions. I can't see anything on the chat, but
there's 2 more questions from Jeffrey Stallman, but they're in the Q&A section.
okay.
So the first one is for Jonathan. So maybe you can just read it yourself there. I think you can also see this Q&A section and answer, Jeffrey. The other one is for Harte. It's also heads up there.
Yeah, I just saw the question. So hi Jeff, thanks for the question. And yes, you're right, and there's a reason for that, because in the slide I showed, I choose to show only the number of species that are considered threatened, along with those that don't have enough data. So it's normal that when you add up all the numbers shown in the slides, you don't get the full total of just under 7,000 species, because I did not include the categories apparently secure, not applicable, secure, and there is also one species that is possibly extirpated. But on the slide on the left, the pie chart on the left side of the slide, those show the proportion of conservation ranks based on the full set of all the assessed macrofungal species. So thanks for pointing that out, Jeff, and I hope that makes things a bit clearer now.
Yes, and I'll answer that other question. The temporary code names, they are predominantly derived from the molecular barcode, being something that has no match in any reference database. And we're using NCBI, everything on iNaturalist, everything on Mushroom Observer, Unite, and additionally some sort of private curated databases where people have basically marked which sequences are reference sequences. And so that's really the primary way that we assign these names. But in some cases, you may have things that look like they're easy to identify morphologically, but are really part of a species complex. And so while they may look like something that you could confidently give a name to, we can see evidence via the DNA that there's most likely multiple species that look the same. Hopefully that answers the question.
I would just make one more note there as well. The temporary code names are just kind of the first step. The next step is actually to start putting provisional names on these things. And then the next step is to start getting them described. And so, you know, in our perspective, a provisional name should be reserved for things that where we have a number of collections, where we think we know something about the morphology and the ecology, and there's someone willing to, you know, take the first kind of, you know, the first step in starting down the path of description. And so that's, you'll probably see provisional names on more of our stuff in the future, but we just aren't there yet. Yeah.
Okay, thanks. I don't know, do we have any more questions somewhere around I can see?
It doesn't look like it, at least. So maybe I can do a little recap, which will take like half a minute. And in that time, if anybody has a question, you are allowed to unmute yourself at this point. So let's start in Canada. It was really nice to see everything that's happening in North America. There is still data lacking there, but with your combined efforts, it seems like it won't be the case for a long time. So it's really, really impressive to see. It's epic that you're doing the theatrical mycology. I'd never heard of it, but the photos, at least they look great. And I wish I would have seen one of these presentations myself. And I look forward to an invite someday at least. Also great visual on this Mycosphaera website with the association networks. I couldn't believe what I was seeing. It's quite futuristic actually to see both trees and all the fungal names attached. So I'm not sure if this is actually already visible for the broad public, but at least I hope one day.
Not yet, but we plan to release it. If not this year, maybe probably next year.
Yeah, I'll keep an eye on it. It looks, yeah, for sure. Just browsing it would be a fun evening activity. Then for FunDive, just I would love to be one day in this, like where these big foray sequencing events, if you're all under one roof. I think it's a really nice way to build a community and actually to teach people also about this molecular method. Yeah, even those that have no background in it at all. And I saw all of those little white boxes full of mushrooms in there, and that's exactly the way that we do it as well. And I can imagine everybody sampling and getting the samples to you. And then, of course, for Stephen, I've been watching your work for quite some time. I'm always impressed when you make a Facebook post and read it with a lot of interest. And I think, yeah, I'm definitely not the only one. It's crazy what you are doing now at the continental level in North America, but before even in Indiana, and I think I'm from Belgium, which is even smaller than Indiana. So if you can do it for an area that's about 3 times as big, I'm sure we can at least make an effort to also make something like a couple 100,000 collections in let's say a decade or so. That would be really, really nice. The bioinformatics as well, the fact that you all share them, it's an amazing resource that I also use quite a lot myself, and not only use but also serve as an inspiration. So I do thank you and your bioinformatics team for that as well. I think we have 2 more questions by now. I'll let Jacob go first, and then I'll stop talking. And then there's a other question by Hira after that, and I think. Yeah, once those are done, we can finish the meeting. Yeah, Jacob, you can go first.
Yes. First of all, thanks for the answer before. I mean, this crowdsourcing approach is definitely also something I want to explore a little bit in a Danish and perhaps European context. But the other question is more related to how I'm just very curious to your thoughts on whether how you see the future for fungal barcoding. Is that something that is centralized versus something happening in each and every field camp you have? Where do you see the balance? I mean, there's pros and cons in both workflows. I'm just very, I mean, I'm in Denmark, we're still doing it pretty centralized, and we do the same in FunDive. But I mean, you could also do it when you are in the field, and I don't know exactly how the technologies are evolving. I know with the nanopore you can in principle do it, but it's maybe not really worth the effort towards the effectiveness. How do you look at that? And of course, it's hard to predict the future, but with the current technologies, how do you see the optimal way to do it?
I guess I can answer from my perspective. Yeah, as far as being optimal, I think distributed networks are a great idea for really high throughput sequencing. There's certain approaches like a hub and spoke approach, and this is something that I'm developing with my private company, where people can do the PCR in their wherever they are, and then send in the samples for sequencing in a larger in a sequencing center with the nanopore. That way people don't have to have 10,000 specimens to barcode at one time in their location. Bringing the gear to the field, it's more a stunt. It's, I think, a way of getting exposure, showing people, being able to train people, and really to teach people that these sort of molecular tools are not locked behind the walls of academia, that it's really something that anybody can learn to do. And the more people that pick up these skills, the more people may be confident enough to enter the world of academia. And the performative aspect of what we were planning on doing at NAMA this summer is to be able to get the time from sampling to sequencing so fast that before the event is even over, we'll have a running list of every specimen that's come into the event. And so again, it's more of a novelty, but I think practicing this process and developing this process, once the technology becomes affordable and portable enough, this could be something that is optimal, where you can equip every major event will have a team that's doing this, and you'll have really rapid turnaround from collection to sequence collection, to the point where you can then decide, okay, which specimens should we take, what's new, what's interesting. So yeah, hopefully that answers some of that question.
It does. Thank you so much.
Yeah, I'd just add a couple things on there. In terms of the future, there probably will be a couple limiting factors in terms of a true broadly dispersed model. And I think the hub and spoke type model that Harte talked about is probably going to what wins the day. From the perspective of currently, we started out using $100 flow cells called Fungals. And they do about 1,000 specimens on a run, and they're discontinuing those. And so that's going to require a flow cell that it now costs $1,000, but you get to do 10 different libraries on that same flow cell. You can wash it and reuse it. The Fungal you can't reuse, but you have to be able to support a very large number of specimens in a short amount of time to make full use of that $1,000 flow cell. And so that there becomes a bottleneck there in terms of just organizing enough specimens to have a run. Then there's a bunch of other things. Just take something like the cost of the tech, the master mix. We buy it in bulk and have it custom manufactured to our spec, and we get it at a fraction of the price that probably anybody else can get it for, because we're ordering in quantity and we're ordering a custom mix. And so that just by virtue of having scale, you gain certain quantity, efficiencies in terms of pricing power. And so, I think there will be a balance in the future of both. but I would love nothing more than for the Ontario people to get up and going. And I've told them I will do everything I can to help them, just because it's too much of a burden. You know, there has to be more distribution than there is, to offload the work in multiple places. And then also there needs to be, um, backup labs, right? Like it just can't be dependent upon one or two labs to do all this.
And that's what we are trying to do.
Super. Thank you so much.
I think Ijaz, I'm not sure how to pronounce it.
Question.
Hello, everyone.
Hello.
Thank you so much for such an interactive talk. And my question is that in biodiversity-rich developing countries, the molecular taxonomy is often limited by funding and infrastructure. So what sustainable models exist to ensure long-term support for biodiversity research in these areas? Like I am a PhD scholar from a developing country, Pakistan. We face many funding problems to document such large biodiversity from the hotspot areas. So is there any open access sequencing platforms or global databases which better support researchers from developing country in contributing to biodiversity documentation?
Yeah, I can answer that because I'm seeking a lot of international grant, but I see a lot of grant that are specialized for development countries, underdevelopment countries, I would say. And so I think it has a lot of opportunity. I would say that I have been approached by the Mohammed Ben Zayed Conservation Fund. That is the only international fund that finance especially fungi conservation. So I've been approached by the board member administrators. So you could check for the Mohammed Ben Zayed Conservation Fund, and everyone here, I tell you the secret. And they are actively funding mushroom conservation. It is small grant and a grassroots movement conservationist. So it is not a scale grant, like it's less than $25 US, $25,000 US. And you can check Grant Connect, and if you tap with AI research on website to see development countries, philanthropic funds, conservation funds, probably you can reach, even if they don't finance especially fungi, because fungi, you can create a plan to try to convince them to finance this. It would be my advice.
Yeah,
okay,
I would add
thank
one.
You so much. Thank you so much.
I would just add one thing to that. So, we're a small lab. We can't offer grants external to our lab or anything like that. But one of the things we have been doing for the last number of years is offering free sequencing services to people in developing countries. We work with 3 different groups in Pakistan at the moment. We have more sequence specimens from Pakistan from our lab than a number of US states. And so, everyone shouldn't need to be dependent upon sending stuff to the US or something like that. And it'd be great to develop local capacity. But until that's possible, we do offer that as a no-cost service for developing countries. And we'd like to help people get their projects going in any way we can. One of the reasons that's really important to us is I mentioned we do a lot of type sequencing from the Field Museum right now. And a lot of those specimens are global in nature. And so we're starting to have more of a global interest at this point.
So what is your lab? Could you please repeat the name?
Yeah, Mycota Lab, mycota.com. I can drop our links in the chat.
Yeah, sure. So what would be the protocols for sending the samples over there?
The same as I mentioned in the talk, which would just be upload them to iNaturalist, dry them out, and mail them in. It's that simple. You just need to put.
Thank you so much. Thank you so much.
You would just need to put the iNaturalist number or a QR code of the iNaturalist number with the specimen. But we try to keep the protocols that simple.
Okay. Thank you so much for the informative talk. Thank you.
That was really awesome. But there's also one more question I see in the chat from Colin. How many specimens are optimal for 1 million flow cell? And that's a question to Stefan. So I also can ask similar. It's like you mentioned, you are going up to 500 specimens? No, even more, right? Or am I correct?
On the Fungal, we did 1,000 specimens per run, 960, so 10 plates worth. And now all of our runs on the MinION are 2,000 specimens, so 20 plates worth. And we typically get 8 to 12 washes from a MinION. So one MinION supports roughly 20,000 specimens to this point. For $1,000.
And is this like, and is this, what is the successful rate of barcoding then, or like sequencing in the end? Is this like, are you able to recover a signal from most of the samples, or there are like some problematic?
So I would say most of the problematic stuff is based on the primers that are being used to this point, and so those will be well known to you, I'm guessing. Extraction doesn't seem to be a problem. We do a second pass on everything that fails, and we'll put a bead in it and bead for that second pass, so that helps with some of the more recalcitrant things. But we've had success. There was one set I posted about recently from the Rocky Mountains in the U.S. It was 600 specimens and 96% success rate. So yeah, I mean, we commonly see success rates above 90% for modern collections. I would say our protocols that I mentioned for the museum specimens, we have some sets of 1,000 for like the agaricales that were over 90% success rate for average 75-year-old specimens as well. And so there are really effective protocols for older specimens as well. But we do have different sets of protocols depending on the age of the specimens. If a specimen's 5 years or older, we'll usually kick it into the museum protocol.
Oh, that's great. That's really, sounds really like successful and really cool. Okay.
Yeah, I think then we can call it a day. Maybe just one more comment to Melody. This will be uploaded on YouTube somewhere later this week where you can rewatch it. And then I want to thank the speakers again. It's been a very interesting evening with beautiful presentations. You've all conveyed a lot of information within a limited time, for which I do like to thank you, and for the questions as well, and then for my co-host Marta for helping organizing this. Thank you.
Thanks a lot
Thank you.
for inspiring talks. Yes, and I can just very shortly announce that we see each other within this webinar next month, and we will have Lukas Picek talking about, I think, AI tools in mycology in general, but this will be announced soon. So we will obviously post this on the website and social media. So thanks a lot, and thanks a lot for sharing all of the protocols and informations, and let's hope we can also share something from FunDive part within the sequencing in the future to North America community. So thanks again, and yeah, should we close the meeting and let everyone
I would
go?
have one question for you, for Jacob and Marta, but it's not private, people can stay, but it's the Uppsala Congress. The Uppsala Congress, do we need to register ourselves even if we have applied for the grant travel?
Maybe I can answer that question. I think you should register. But you really need to contact the Swedes directly, and do let me know. Send an email to me if you need the more exact context. But
perfect.
I mean, from FunDive, we are only co-organizing, so it is really the SLU that are in charge.
Right.
And I'm afraid the deadline was yesterday, but I still think you can make
tomorrow.
it. It's tomorrow. Okay.
Yes,
Yes,
because
tomorrow.
I still need to register, and it's tomorrow.
Oh, super. Thank you.
Yes, 25th of February. Yeah, that's for people who are not aware of what we are talking about. In April, I think it's April, I don't remember, 14, there will be a conference dedicated to fungi conservation efforts and actions in Europe, I think, mostly, right? So I can also post the link in the chat about the conference if someone will be interested. Yeah, so the deadline is tomorrow, but I think we we can close our webinar and see with everyone or people interested next month.
Thank you, Marta. Thank you, Jacob. Thank you, Glenn. Thank you all. It was a pleasure.
All right. Have a good
Yeah,
night.
thanks for having us.
Yeah, thanks a lot.
Yeah,
Have a
thank
good
you
night
all.
and good
Bye-bye.
time. Bye.
Bye.
Thank you. Bye bye.
Thank you all. Bye.